This is the dissection of the sheep brain, and as we go through the dissection, remember that sheep walk on all fours, humans walk on 2 feet, so their head's going to be projected forward compared to ours. So when we're looking at these cerebral hemispheres, you note that there are convolutions, or these lumpy bumps with nooks and crannies. The lumpy parts are called gyri, G-Y-R-I. The indentations are called sulci, or sulci, S-U-L-C-I. And this long separation right here is called the longitudinal fissure. When we move back here, this is the cerebellum, and this is the brainstem that's projecting off of the back. Right now we're going to turn our sheep brain over, and we're going to take a look at the structures underneath. First, we've got these olfactory bulbs at the anterior. You can continue to dissect those out if you wish with a scalpel or with your probe. Sometimes you can make your way along the edge and loosen it up. These olfactory bulbs sit on top of the cribriform plate of the ethmoid bone to pick up the sense of smell. As we move posterior, we see this X. This is called the optic chiasm or optic chiasma. Both are correct. But if you can imagine, these are the optic nerves that then move forward to each of the eyeballs. But what they've done is they have cut these. So what we're looking at is the optic nerve from the sheep's right eye moving towards the occipital lobe on the opposite side, and the sheep's left eye crossing over to the right side of the brain. And that crossing right there is called the optic chiasm. I'll put the probe in it so that you can see it. As we move posterior to that, we come upon the pons, the medulla, and the spinal cord. And the way that you can remember those in order is PMS: pons, medulla oblongata, and the spinal cord. We're going to turn the sheep brain back over, and we're going to perform a sagittal incision. Sagittal means that we're cutting it into right and left pieces. So this is a mid-sagittal cut where we are going to divide it into even left and right pieces. Make sure that you cut cleanly through. If you have a partner with you, you can hand half of the brain to your partner for examination. This is one of the cerebral hemispheres. When we look inside, we see this white area. This white area here is called the Corpus Callosum. And the Corpus Callosum connects the right and left cerebral hemispheres. We can put our probe just underneath that and just move it back and forth. And we can open it up. That's part of the ventricle system in our brain. The lateral ventricles are called lateral ventricles because they move laterally through the brain, which means off to the sides. You can put your finger in there if you want to do a touch and feel. But when you are looking at your human models or your diagrams in your textbook, don't forget that these lateral ventricles continue on laterally. They're not flat. So the cerebral spinal fluid is created in the ventricles. It is high in glucose, and it is made by the ependymal cells, which are the central nervous system neuroglia that are ciliated. They create cerebral spinal fluid and also circulate it. We continue on with our pons, medulla, and spinal cord. And as you can see, our cerebellum is attached to the spinal cord. If we pull that cerebellum back, we'll see a set of lumps. When we put those together, there are 2 large lumps at the top. Those are called the superior colliculi, and then 2 smaller lumps underneath. Those are called the inferior colliculi. And colliculi control your emergency response of your eyes and your ears. Together, they're called the corpora quadrigemina. And whenever you have an emergency situation, you automatically turn your eyes and your ears in that direction to protect yourself. But when we look at this cut cerebellum, you see this whitish tree-like structure? This is called the Arbor Vitae, or the Tree of Life. We're looking at the alternation of gray matter and white matter. And as you can see, gray matter is not really gray, and white matter is not really white. But this white matter is kind of a creamy white color, and it is showing us where the brain has been myelinated. Myelin sheath is a fatty insulator, and we do not have any dendrites in that area. Dendrites are only in the gray matter. It doesn't make sense to put dendrites in the white matter. We don't want to insulate dendrites because then we wouldn't be able to pick up information into the nerve cells. This myelin sheath in the cerebellum is very important because it helps to direct our information. The cerebellum is involved in balance and equilibrium. And for patients with multiple sclerosis, they undergo demyelination of the arbor vitae, and this can send electrical signals that are not correct. If you have the patient bend their chin to their chest, sometimes they'll complain of an electric shock sensation going down their spine, and that's because we have lost that myelin sheath there. So let's go ahead and take that cerebellum off of the spinal cord here in the brainstem, and it is attached by tissue called the cerebellar peduncles. We're going to just remove that, and you can hold that in your hand and take a closer look at that arbor vitae. We can turn it around this way, and you can see that tree-like structure, the arbor vitae of the cerebellum. So in review, we have the arbor vitae of the cerebellum. The cerebellum is involved with balance and equilibrium. We have two cerebral hemispheres, the longitudinal fissure in between, with the gyri and the sulci, the corpus callosum, and part of the ventricular system, that's the lateral ventricle. We also have our pons, medulla, and spinal cord, which can be seen from this perspective also. Pons, medulla, spinal cord. This is the optic chiasm. And finally, these are the olfactory bulbs.
Mama.
And that's it for the sheep brain dissection.